information in the chat about potential future seminars. We have a couple of tentatively planned seminars on NMR that we'll be hosting over the next month or so. We don't have dates set for those, but once we do get them, we will post that on the website in the hopes that you'd all be interested in attending that as well. So with that, I will go ahead and get started. I'm pleased to introduce Dr. Prithwiraj D. Raj, as we call him, is a chemist by training. So he received bachelor's, master's, and PhD in chemistry from University of Calcutta and University of Kalyani in India. Served as a postdoctoral fellow in 2008 to 2011 in France, working on design and synthesis of anti-TB and anti-cancer drugs. He joined the CSU microbiology department after that, working in oligosaccharide characterization, glycan characterization, and assay development, focused primarily around Mycobacterium tuberculosum, TB antigens. In November 2021, he joined ARC and is contributing actively to our metabolomics and small molecule profiling areas, particularly in GCMS. And today he's going to present to us some of his knowledge and skills on the use of mass spectrometry for glycan characterization. So with that, I turn it over to you, Raj. Thank you.
Thank you, Corey. Let's begin. Glycomics has two parts, glycochemistry and glycobiology. Today, I'll be focusing on the glycochemistry. Glycobiology is an essential or integral part of it. It's another phase. While biology acts as the source of glycans and carbohydrates, chemistry does the structural analysis, give the absolute structure back to biology to understand the biological system. That's how it works in a synergistic manner. And what kind of relevance with the absolute structure one can establish? I wrote some wild example, like deoxyribonucleic acid. If we change to deoxyxylonucleic acid, what molecular manifestation happens? This transformation is basically just one single transformation using C3 epimerase. So these sort of things come. Nevertheless, let's talk about mass spec and carbohydrate, and which is pretty difficult practically because carbohydrate is full of stereoisomers, same mass spec fragmentation pattern. That is why it's kind of pretty difficult by mass spec to distinguish the different monosaccharides and different pyranose, furanose conformations, as well as enantiomeric forms like D and L, etc. Let's understand a little bit more about this. Let's have these two molecules. Say we have galactose here connected 1,6 in alpha fashion with the glucose, and here we have one glucose at a similar fashion with another glucose. Presently, mass spec or MSMS will give the same MSMS fragments. So annotation by a database or reference is pretty difficult. It's pretty confusing. Both the structures will come with the same score, and it is impossible to distinguish which molecule we are actually after. So it needs chemistry or human intervention. Why? Because monosaccharides are stereoisomeric. Here you see the D-aldohexose families. Aldo comes from this aldehyde group on the top of every monosaccharide, and it's 6-member. That is why it's hexose. And you see there are 4 stereogenic centers. So that carries on in every other molecule. And if and mass spec has no way to distinguish between glucose to mannose or aldose to allose or glucose to galactose to say. And if on a funny note, if one wants to remember the names of all aldohexoses, one can go by this mnemonic. All altruists gladly make them in gallon tanks. And there are some glycan databases like Uniqurb for human saliva glycoprotein. Its mobility is also stored in database like Glycomob. NIST has a human milk oligos, but there are 9 billion species, and every species, every organelle, every type of cell have different carbohydrate manifestations. There are glycoforest as a software as well, which go for de novo, but it needs manual reference. And Skyline is there also, but it's mainly used for proteomics, where each building block is different. Alanine is different from glycine. Alanine is different from serine or tryptophan. So annotation is much more easier. Now let us see how we detect or characterize monosaccharides. We take a monosaccharide, we just treat with methoxyamine in our routinely in our laboratory, and then treat with MSTFA TMS, which actually put TMS groups on the hydroxy groups, and then we run on GCMS. We see two peaks because this imine has a tautomeric effect, or it undergoes tautomerism to give the enamine. So we see imine peak as well as enamine peak. While enamine is coming late because it's more polar, a nitrogen acquired a hydrogen becoming more polar, and that's why it's looking late. Now, if we have a mixture of sugars, these peaks might interfere with other sugar signals. Although this is a very short and easy reaction sequence, entire reaction sequence analysis can be done in 4 to 5 hours max. So to make it one sugar, one peak, there is a protocol we often follow in polysaccharide characterization, where we need to understand the monosaccharide composition. We take polysaccharide, we treat with 2 molar TFA, acidify in the acidic condition, all the glycosidic linkages will break up, make it into monosaccharide, and then we treat with NABD4, that is borodeuteride. And then we go for acetylation to put all the acetyl group in all the hydroxy group. Why we use deuterium? It differentiates the reducing end, and it differentiates from natural alditol. In a biological sample, there could be natural alditols, and putting the deuterium will differentiate from which is coming from sugar or which is coming from natural alditol. And what happens? This is a GCMS spectrum. You see the bunch of pentoses, arabinose, xylose, ribose, and you see mannose, glucose, galactose, all each sugar, each peak, one sugar, one peak. We also have myoinositol. Just to note that myoinositol is not a sugar, but it has a sugar-like structure. And you may see that rhamnose and fucose are also coming along with the pentoses, although they are hexoses, because they are deoxyhexoses, and one hydroxy group at 6 position is not there. So they behave more towards pentoses on GCMS. Now, we understand the D and L monosaccharide. This is D glucose and this is L glucose. You can see these are mirror images, and non-superimposable mirror images, because out of plane rotation of the molecule is not allowed by stereochemistry law, the rule of stereochemistry. So they become enantiomers, and these enantiomers pose enormous challenge to analytical chemists to separate, mainly separate, and then characterize. So we see how we can separate them and then characterize. And to do that, we'll go to their cyclic structure. We have D-glucose. It goes for a 90-degree knockdown, goes for C4-5, this bond, C4-5 bond rotation, and then goes for cyclization. Now, this aldehydic carbon being sp2 hybridized, this is a planar structure. This carbon is planar. So this hydroxy group can go for a nucleophilic attack both from the bottom and from the top, giving rise to two different conformation. One is alpha, where hydroxy group is bottom and CH2OH group is at the top, and beta, where is hydroxy group and CH2OH group on the same side. Now, as soon as we did this cyclization, we generated one more chiral center. That's a new chiral center, and that is why alpha beta 2 conformations are obtained. And these anomers are diastereomeric. So as soon as it is diastereomeric, it is not enantiomeric. These can be separated by a chiral chromatography. And L-glucose also goes for these, but if you see L-glucose, alpha L-glucose and alpha D-glucose, they still maintain the mirror image relationship. So, what do we do with these? Can we use them? Note that another factor is that all steps are in equilibrium. If you leave alpha glucose in solution like in water, over the period of time, you will have a mixture of these three forms: linear form, beta form, and alpha form. So this factor, this phenomenon is known as nutrarotation of sugars. This happens in aldoses as well as ketoses like fructose and stuff. So how can we separate alpha beta anomers by chromatography? Yes, we have to tune a little bit. We increase the column length, like you see here, 50 millimeter C18, 5 to 95% acetonitrile water has been changed to 150 millimeters. So increase the column length as well as the buffer condition to separate. Okay, we separated two forms, two anomeric forms, alpha and beta, but we don't know which one is what, which one is alpha, which one is beta. To do that, there is no option but to go for NMR. And if we do decoupled, get a decoupled carbon-13 NMR, we see the peaks like this, where early looping NMR having coupling constant. Coupling constant is the difference between the two peaks multiplied by the magnet power. So it is 161 hertz, which corresponds to beta glucoside, and 171 hertz, which corresponds to alpha glucoside, which is late eluting NMR. And if we have glycoside, we can straight away go for simple proton NMR, where we can see that alpha NMR is characterized by the coupling constant of 2 to 3 hertz, whereas beta NMR, little downfield shift, that is shift on the right side, which gives 5 to 7 hertz as coupling constant. So we need NMR to characterize. We need NMR to characterize the NMR. Now, how can we use alpha beta conversion in MS? By locking conformation. How do we lock? We lock by putting this R group, alkyl group, with the anomeric hydroxy group, and that way we have two anomers, which name has also changed as glucopyranoside and as alpha D glucopyranoside and beta D glucopyranoside. Why we need to change that? Why we need to put alkyl group? Because of this mechanism. This proton is responsible for ring opening. So we remove this with carbon or any other atom, and that locks the conformation. Now, what happens? For example, we have arabinose. We open chain arabinose in solution existing all these 4 conformations. And what happens? We do put any alkyl group, which is chiral. Chiral means there is a stereogenic center. We get here, we put R2-octanol with that. So we lock the conformation in a chiral manner, and then put trifluoroacetyl group on these hydroxy groups to make it amenable for GCMS. And we see the 4 peaks in GCMS by SRM, single reaction monitoring. And these 4 peaks correspond to all these 4 anomeric conformations. And NMR shows that 2 are beta. You see, this is beta, and this is alpha, this is alpha. And the ratio of the peaks is almost similar to what we obtain in GCMS. So we could detect alpha and beta by mass spec with little help from NMR and chiral glycosylation. But the question is, do we separate D and L isomer? Yes, we do. In this process, L isomer gives a peak pattern like this: 1 peak, major peak, and then 3 others here, whereas D arabinose coming as 4 almost equidistant peak, and you can easily distinguish between D arabinose and L arabinose. Then we come to linkage or connectivity. Now we know how we detect the monosaccharides, how we detect D and L. If we have done that, we move to the oligos, where different sugar units are attached to each other. And because there are positions like 2, 3, 4, 6 in a hexose, any hydroxy group can bear any sugar or any substitution. So we need to know where exactly it is connected. Like here, it's in a beta fashion as it is drawn. In 1 to 3, this one position is linked to 3 position of the other sugar. So the one position of this sugar is referred as non-reducing end, and the 3 position of this sugar, or this sugar practically, is known as reducing end. Now, let's see how we determine the linkage via permethylation and using GCMS. Let's see this molecule. We have a hexaarabinose, arabinoside. This is part of a molecule which is a polysaccharide of 15 to 17 kilodalton from mycobacteria, and it has 4 kinds of arabinose. It has 2 terminal arabinose, 2 arabinose, 2 linked arabinose. This is 2 linked. This is also 2 linked. This arabinose is 3 and 5 linked, and this is simply 5 linked arabinose. Now, when we do permethylation, all the hydroxy groups which are free, which are not connected, will be transformed into methoxy groups, and then we release them as monosaccharide. So what happens? The hydroxy groups which were connected with glycosylation, except 1 and 4, as you see, 1 is always connected and 4 is always connected to form the ring. So except 1 and 4, so 2, 3, 5, any one is connected to other glycosidic linkage, other sugars, those will be acetylated. So like tiara, we have 1 and 4, fine, they will be always acetate, and none of the other, like 5, like 3, like 2, they are all methylated. That means they were all free. So we have a specific molecular weight, specific mass for terminal arabinose. Similar fashion, 2 arabinose. Now, 2 arabinose, and then this is 3, 5, 3 and 5 are acetylated. So this is 3 and 5, and this one is simple 5. So we'll have a spectrum, GCMS spectrum like this, where we have 3 arabinose, 2 arabinose, 5 arabinose, and 3, 5 arabinose. You see other peaks, mannoses, because they come from the rest of the molecule. And we have these peaks in well resolved, and they have specific mass fragmentation like this. I'm showing an example for 2 arabinose. If we draw the structure and break the molecule in these bonds, we will see that these values correspond to the GCMS mass fragmentation pattern, and where 190 corresponds to this top part using this one, and 161 corresponds to the bottom part using the upper 2 carbon. Now we have this, and which correspond to exact mass, and one may ask that you have the major ions at 129 and 130. These are further fragmentation of losing acetate or acetic acid at 60, loss of 60 Da, and this 161 losing a methanol with 32 Da. How do I know this is methanol or acetic acid? Because we know, because we have derivatized with acetate, and we have derivatized with making methoxy. So that's how it corresponds, that this peak or this spectra correspond to 2 arabinose, 2 linked arabinose. Now, to summarize what I have just told so far, there is a beautiful work from Professor Seeberger's group, one of the finest glycochemists in present day. And this is a synthetic oligosaccharide where they make this trisaccharide, and they change these sugars sugar. This changed to glucose and galactose, and which also changed the configuration of this particular linkage to alpha and beta, and produced six oligosaccharide. And they tried on ion mobility MS, and you can see on this side that alpha versus beta is well resolved in both cases, 3 and 2. 3 is this guy where it is alpha galactose 1,3, and 2 is beta galactose 1,3. So same linkage 1,3, but alpha to beta they are resolved. And then comes to 6 and 5. 5 is beta 1,4, another position, and alpha 1,4. So here the alpha to beta change is happening, keeping the 1,4. Again, they are well resolved. And then 1,4 versus 1,3, 6 and 3. So this is 6, alpha 1,4, galactose 1,4, and alpha galactose 1,3. So they were well resolved. But when it comes to glucose 2, like 1 and 2, you have beta glucose 1,3 and beta galactose 1,3. Mass spec cannot separate these things. So this is the limitation where mass spec comes to a knees, that they cannot distinguish between the sugars. Why ion mobility was chosen here? Because ion mobility uses gas phase mobility of the ions based on their mass, charge, size, and shape. Here, mass of all the oligosaccharides were the same, 589. They were being measured at n-h, 588. They have almost similar size, but the alteration in connectivity and stereochemistry actually changed their shape, and that reflected upon the separation or different CCS values. Here, CCS values are entirely dependent on these 4 factors, and which is a very crucial and another dimension along with mass, charge, and size that gives the molecular characterization much more feasible and easy to separate and distinguish the stereoisomers. Let's move on to tandem MS fragmentation. To understand this, the most crucial thing in tandem MS for oligosaccharides or polysaccharides is ionization, because it is not like proteins where a lot of nitrogens are there. They can be easily ionized, and then subsequent fragmentation will take place. Here, 2 aspects is that there are slow ionization like CID, which my favorite, and there are fast energy ionization like ETD, electron-mediated dissociation like ETD, ED, and UV photo dissociation. UV photo dissociation has a restricted applicability because it needs chromophore to initiate the ionization. Whereas these are developing, still under development, this positional dissociation of high energy electron, DH, CD, etc. But thing is that for an analyst of structure, you need to have the fragments which are useful should be carrying more chemical information rather than only the sequence. It is not only about the sequence of the glycan, but also the stereochemistry and related like post-translational modifications. All those information will be lost if we go for high energy dissociation. Let's see how they differ in fragmentation pattern. This is a oligosaccharide with 2 glucanac and 5 mannoses, just proton M plus H in CID. It's a minimal fragmentation with just losing the sugars and giving the sequence. Where it goes for with salt, salt induces further fragmentation. With Na, sodium, it gives much more fragmentation. It gives much more fragmentation, much more information. And when go to negative mode with phosphate adduct, it gives the ring cleavages, cross-ring fragments. And when we go to ECD, high energy stuff, its molecule is almost decimated into small peaks, and so does in UDPD. And these are nightmares for any structural analyst. It's very difficult to pull out proper structure that way. Now, how we do then glycan structure elucidation? Three things are very much needed. One is to identify the number pattern, because output of mass spec is just numbers, nothing else. Then analysts have to identify the number pattern. So you have to use you have to be intuitive. I owe my intuitive nature to these two particular gentlemen. I won't elaborate on those, but then it comes to justification of the fragmentation mechanism. These two things govern this mechanism. One is bond energy and product hand stability. And third thing is then using this information, you have to paint your absolute structure or construct your absolute structure. Let's do it to elucidate this particular structure, how we do it. Now, if we do monosaccharide analysis, we will get mannose and N-acetyl glucosamine or glucnac in 5:2 ratio. And then if we do the linkage analysis, we will get 3 T-mannose, 1 T-mannose, 1 T-mannose, 1 T-mannose, and then 2 3,6-mannose, this and this guy, and then 2 6-linked glucnac, these guys. But what we derive from this information, and if we do NMR, we'll get that all the linkages are alpha. So conclusions that can be derived from these three information is that one, no glucnac is at the non-reducing termini because no T-glucnac was present. So none of the 2 glucnac can be written or can be drawn on this side. That's a restriction. 2 out of 5 mannose, 3 are T-mannoses. And so there are multiple branches, definitely. 2 or 3 6-link mannoses indicate branchings are at 3 positions in 2 of the 6-link mannoses. These are primarily 6-link mannoses, and then 3 positions they have branching. And the backbone of this molecule is all alpha 1,6 liposylated. Now, how do we do the sequence? To do the sequence, we need to do MSMS. And why we need MSMS? Because these are the conditions restriction we impose on our molecule that these should fit these conclusions. And all 3 structures, there are more structures possible, but the way we'll be elucidating structure, those rule out those structures as well. So these 3 structures also fit all these bills. Let's see what we do from MS. Now we have this structure that we have our target. We have these alternative structures as well. We see simple M plus H CID. What first thing we see? We see that this molecular ion peak is actually fragmenting in 2 pathways. First one is losing at 221. What is 221? 221 is the intact glucnac. Now we know glucnac cannot be a terminal sugar. It cannot be a non-reducing end sugar. Cannot be in the middle because the entire molecule is intact. In the middle, you have to break in 2 parts at least. So it must be at the reducing. As soon as we put that at the reducing end, we eliminate these 2 structures. These are not possible. Now this is still there. And then we see the other pathway. It loses all mannoses. 1, 2, 3, 4, 5. 5 mannoses are gone. Then what we see? We see a 425 peak, which is 2 glucnac units. And then it releases it goes for 1 loss of 1 glucnac, entire glucnac, and goes for 204. So this clearly indicates that 2 glucnac units are together, side by side, which makes this structure invalid. So we have this structure now. Now, I may say that, okay, you elucidated the structure, but tell me why you see the B fragments and not the Z fragments like this. This can also be 204. Why you are telling that this is 204? Because you see the linkage here is 1,6. This bond is a primary alcohol. So as soon as it breaks in this fashion, which is Z fashion, it has to produce an exocyclic double bond. Now, exocyclic double bonds are not stable. So product ion stability governs the fragmentation. You don't see Z fragmentation because it was 116. Now there are other fragmentations as well. You see, I was talking about B fragmentation, which the reducing end will have the corresponding Y fragmentation. C fragmentation will have the corresponding Z fragmentation, and A and X fragmentation are normally the cross-ring fragments. Here I have shown the Z fragmentation where it happens when secondary alcohol, that is 2, 3, and 4 are linked with other glycans or glycan part. This can happen, and I will show where it happened exactly. And I have also illustrated here that these guys are non-reducing. They don't have non-reducing end because you see the anomeric carbons are linked via glycoside linkage. So with each other, so trihalose and sucrose, they are not reducing sugars. Now, what is cross-link fragments? Let's take the example of this guy where 1, 5x, this bond is 0, this is 1, this is 2, this is 3, this is 4, and this is 5. So 1, 5 fragmentation is happening. 1, 5 fragmentation is happening and producing this diene and this carbonate. This is exactly retro-heterodyne Sandler reaction. This is 4 pi and 2 pi system giving this Sandler product. You may say that where is the double bond? See, non-bonded pi electrons are present in the ring, which is very much involved in all anomeric reactions that sugars carry out. There is alternative mechanism. A traditional mass spec analyst will tell that, oh no, there will be this kind of this kind of either oxygen carrying charge or this hydrogen is cleaving by a homolytic cleavage, where 2 electrons goes to the either atom equally, and then go for this kind of fragmentation. But if you follow this kind of fragmentation cannot be explained, because if this hydrogen is replaced by a substitution or carbon or any alkyl group, then this fragmentation pathway cannot follow. It has to be considered like heterodyne Sandler reaction. Now, does MSMS talk to stereochemistry? This is a huge question. If you ask any traditional before who are used to analyze with old mass specs, they would say no, you cannot connect to stereochemistry with MSMS. Now you see, we have here this particular connection where this is alpha and this is beta, and I am showing this this fragmentation happened to alpha and not the beta. Why this happened? Why it is not breaking this to give this? You see, when the product is forming after breaking, this is a trans diol. And when the product is forming after cleavage of these beta arabinose, it is forming a cis diol. And in cis diol, the lone pair repulsion between the 2 oxygen won't let it be stable product. So product ion stability governs the mass spec fragmentation. You may get these fragments, but only at high energy. If you put high energy, you will lose this information of alpha and beta, and get the fragmentation of within 90 and so. So, but this needs more work to generalize this concept and include in any sort of algorithm that may come up for de novo characterization of oligosaccharide. Now, there is also a concept of tagging where, but there is a caveat. You should not use other than homopolymer. Homopolymer means same sugar making the entire oligosaccharide. And usually 2-aminobenzoic acid or 2-aminobenzamide are used as tags for this reducing end and produces C base, and then we reduce with cyanoborohydride and stuff like that, borohydride and salt. So we can label the reducing end and then we go for MSMS. This has to do with characterization where to start the MSMS fragmentation or how to find the pattern of MSMS fragmentation because there was a tag. But do not use while we should not use this when PTMs are there, when these post-translational modifications are there, because this reaction actually destroys all the ester groups and all the labile functionality one molecule may have. Now, going into post-translational modification, simply speaking, it is best for post-translational identifying post-translational modification and their location. NMR is the best option. Yes, if you do a 2D NMR, you will get all sorts of information, right, from which type of post-translational modifications are present, like acetyl, succinyl, butyryl, octyl. All these information will be there. There will be location, possible location of those, like C6 is carrying most of the acyl groups, and one of the C2 is carrying one of the acyl group. Also, because this is methoxy groups are there, the 6 positions are methoxylated. All these sort of information can be easily visualized in NMR. And to further investigate, one can do stuff like nosing, and there you can find which environment you have to look for this particular group, functional group. Like octyl here is characterized at near to alpha-1,6 linkage, and in this entire molecule there is only one alpha-1,6 linkage, so it has to be on the towards the reducing end. So this sort of information is clearly and easily can be identified from NMR, and so then you go for MSMS fragmentation, and you can go for your fragments and find exactly which sugar this is carrying. These all fragments correspond to this particular place for the octyl, and these acetyl groups and the butyryl groups are all on C6 positions. Now, when there is no NMR, what to do? Yes, it's challenging, and you have to be intuitive. So suppose we have a monocyte, and acetyl group will increase the molecular weight by 42. Now, 42 may be increased by 2-hydroxyacetyldehyde as well. So you have to consider the both possibilities or all possibilities, whatever is feasible within that mass, and then look for that specific cleavage. It can give a specific alpha cleavage with loss of 28 and giving the methoxy as the product. So you have M plus 42 minus 28, and that will be your mass for this guy. And for this, if alpha cleavage happens, it will happen here, and it will lose maybe 15, or it should be very weak. At high energy, it can also lose 28, but it has to have double cleavage to lose that. But it has an easy way, like chemical intervention, is that you treat these to find what is there, treat with mild alkali. With mild alkali, ester linkage will be knocked off and ether linkage will prevail. So You do alkali treatment, come back again to the mass spec. If it still persists, design, you have an ethyl linkage. If it goes to M, that means it was acetate. And then if you have a complex one, when you have a complex one like this one, acetyl to hydroxybutanoate, to get this fragment and work on all possible fragmentation, like losing as ketene, losing as acetic acid, losing as ketene, losing as formaldehyde due to alpha cleavage, propanaldehyde as alpha cleavage, all these sort to establish correctly and unequivocally that this is this residue. Now, bond energy has enormous implications in mass spec. This is an example which is very close to my heart. This is in MTX where xylose was carrying a sulfoxide, MSX actually. The xylose was carrying a sulfoxide. And this is how the fragmentation took place. We see the major peak at 9 to 23, which is 94 Da. And this guy is the 94 Da involved in the sulfur. How it happened. Now, we have the O minus, it was done in negative ion mode. It has O minus, and sulfur has a vacant 3D orbital, which will take the electrons anyway and make these cyclic form. This is a cis octalene form, and cis octalene forms are not stable. It's bound to collapse because of the 1,2 diaxial interactions. And it collapsed into a stable product, which is 923, and this carbon intermediate, which again collapsed because the carbon sulfur bond is weaker, 259 kilojoule per mole compared to oxygen sulfur bond, making this molecule. So this shows that bond energy has enormous effect in mass fragmentation. Yes, this is not directly sugar fragmentation, but it is sugar involving fragmentation. Now, a question is that if it was arabinose with sulfoxide, would the same fragmentation happen? No, because this bond is rotationally restricted, and this is in opposite side. So this octalene formation cannot happen. It cannot be, I cannot finish without talking about glycoprotein. And for Glycan characterization in glycoprotein, one has to take bottom-up workflow, where you take glycoprotein, you treat with enzymes like trypsin, which is very specific because it cuts the lysine and arginine. And or else you can take pronase and procare, which is much more generalized, which give much more generalized fragments. And then you have glycopeptide, then you go for separation, to get enriched glycopeptide, and then you go to mass spec analysis and database search for the glycopeptide. But sugar characterization is not possible this way, because it will give as only neuraminic acid, and other things will be coming as hexose or hexnac or sort of like that. Hexnac can mean glucnac, can mean galnac, galactose, can mean mannac. Anything is possible. So to do that, one has to go for GAS, which cleaves the N-linked glycans, and for the O-linked glycans, one can use reductive beta elimination or O-glycosidase, which is a little specific. For galactose, I remember, as far as I remember, and then release the glycans, then when the glycan pool, then go for derivatization and go for MSMS. It can, for some proteins, there is majority of the proteins, I would say, one analyzes the glycans are in a database, and one can match the database and find the glycans. But for new protein, one has to go for entire sequence of glycan characterization to establish that what sequence or what linkage and which sugars are present. And you can see I was telling about the ionization possibilities. Yes, as we increase the higher energy, the more and more sequence coverage comes. So this experiment was done on orbit track fusion, where multiple ionization methods can be employed. Just to give an example. And there is also another method for glycopeptide characterization, that is top-down method, which analyzes the entire protein molecule by mass spec. But there are problems particularly for glycans, because glycan sequence and those things cannot be characterized. Further problem is that overlapping peaks are possible, which needs a very high resolution mass spec. So that's the kind of method one has to employ for glycopeptide. And to conclude, identify the monosaccharide, identifying the linkage by permethylation, identifying stereochemistry of glycosidic bond, NMR, identifying post-translational modifications, elucidating sequence using product stability, bond energy, and fragmentation principles, combining all with chemical justification to get absolute structure. Without chemical justification, no fragmentation is valid, and it is not any kind of contribution to any future algorithm one may design, computational chemists may design for de novo characterization of oligosaccharides or polysaccharides or glycans. And knowledge of NMR really helps. I would like to acknowledge, obviously, Professor Chatterjee in MIT CSU and lab, particularly because she trusted me that I could do this kind of work. And of course, the lab of Professor Karen Dogos, where most of the molecules I investigated came from, and also CIF, or presently ERC, has been the backbone for all these spectroscopy that I did. And that's all I have to say. Thank you. And any questions you have?
Thank you, Raj. That was great. And even got a Sherlock Holmes reference. I love it. Open it up for questions. Quiet bunch.
Hang on. I have a quick question, Raj. That's a great seminar. Thank you. You know, these mechanistic fragments that you get, are these stable? You know, the ones where you have the small components coming out after fragmentation, or there has to be some conditions for that on a mass spec, or you just do it by your logistics?
No, there is no logistic except the right side of the brain. So it has to the chemical stability has to come from the knowledge of molecules and where it can be stable. The relative conjugation, as it is producing double bond and pi electrons are there, relative conjugation will give the stability that one has to know. That to assign that particular structure to that particular number, and also one has to see that no other possibilities are possible within that number and the structure.
So, you know, it's very interesting. So you have to have some pre-knowledge of the structure, right, before you go on to the platform of mass spectrometry.
Of course, you have to have which kind of sugar is required.
Right.
It is required. Which kind of linkage are required? What kind of stereochemistry is present between glycosidic bonds? Those are also required. And,
correct.
Without that, you cannot directly go to mass spec for glycan characterization. Okay.
Yeah, I really like the alpha beta fragments that you showed, that one doesn't give and one gives. That's why I was asking that you have to have some pre-knowledge based on NMR or whatever analytical techniques you use prior to going on to these fancy fragmentations, right?
Yes. It is about knowledge of the molecule and the product stability, because you design or you give your mechanistic pathway or describe your mechanistic pathway based on product stability and bond energy calculations or knowledge. These are new things. These were not this type of fragmentation were not there before me. And these are crucial for any future development of algorithm, because computational chemists are sitting there to transform these mechanisms into algorithms for specific cleavages for
Right.
specific sugars.
Thank you.
I see a hand up from Claudia.
Hi, Raj. That was
Yes.
an awesome seminar. Thank you. I'm curious about the differences in derivatization methods for like GCMS separation versus ion mobility separation.
Yes.
Is there a rationale for using different types of derivatization for those two platforms?
You have seen the example I have shown for GCMS is that the molecules have to be compatible. So all the hydroxy groups have to be protected or blocked with acetyl groups or trifluoroacetyl groups. But in ion mobility, you may not need those. You can go with the bare sugars as it is. If they vary in stereoisomeric stuff, which considerably changes the structure, you will be able to get it.
Okay, and do you think the system we have here in the ARC would be applicable for those types of studies?
Exactly. Those particular study was done on T-wave synaptic G2S, which is in our bio laboratory we have.
Yeah. Okay. Great. Thanks.
I'll jump in with a question. You mentioned algorithmic interpretation of MSMS. There's obviously been a lot of tools developed around that for small molecules, and one of the limitations for them, even with all of the data that we have available, is there's not enough standard spectra to train these models well and test them. Are there large libraries of oligosaccharide standards, authentic standards, that could be used to build such MSMS libraries for training?
I have mentioned about Unicarb and Glycomob, which is ion mobility variation of Unicarb, which are specific for human saliva glycoprotein related oligosaccharides. Then NIST has developed this database for human milk oligosaccharides. And so these are the databases. And then there is de novo softwares like Glycoforest and stuff. Now you have to understand there are two major theories operate when you go for predictive tool or de novo type analysis. For small molecules, most of the softwares use the force field theory. Force field theory is based on Newtonian physics and are characterized with a rigid bond structure, which perfectly fit the bills of small molecules. But where conformational flexibility comes into play, one has to use the DFT, density functional theory, and quantum mechanics principles. So I don't know how many computational chemists really build on those aspects.
Yeah, there's certainly some efforts in using both of those techniques, but there's actually others that are completely, I don't want to call them ignorant, but it doesn't actually utilize chemistry at all. It's more of a pure machine learning type approaches. And the limitation with those, obviously, sometimes they benefit from some knowledge, and that's one limitation. But an additional one is just you need a really, really big number of spectra to train those models. And there's not enough of them out there. And so the question is, are there even enough purified structures out there by which to generate more MSMS spectra? And I'm not aware of any, but I wanted to see if you were.
No, practically, I don't know. There is a lot of buzz around GNPS also. That is also a similar kind of like Skyline and Glycoforest and a little bit of Unicarb mixed together. That is also another platform, but those are good for small molecules, not for where the stereochemistry and conformational flexibility comes into play. Those are good for proteins because the building blocks are uniquely carrying unique masses. So sequence analysis with Skyline is a perfect tool, near perfect. Nothing is perfect, but near perfect tool. But for glycans, where all the sugars, all the hexoses have same mass, give similar mass, almost equals mass spec MS fragmentation. It's impossible without prior knowledge how to do it.
Other questions? Okay, obviously, if you do have any other questions, you can always reach out. Raj is very available. Thank you again, Raj. That was a fantastic seminar, and I really appreciate your contribution. Just a reminder to everyone, we are planning to have 2 NMR seminars coming up through ARC. You can find that link again in the chat. If you have interest in using NMR here on campus, I think one will be a software, one is a little bit more interpretation, if I recall. So thank you again for attending, and thank you again, Raj, for presenting.
Thank you
Bye,
very much.
everyone.